Good day to all and welcome. I'm Charles Mulligan, Deputy Director of the Comprehensive Cancer Centre here at St Jude Children's Research Hospital. With my co-host Charlie Roberts, I'm very pleased to welcome you to the Spring 2021 Science of Childhood Cancer Lecture Series. It's been truly wonderful to see the enthusiasm for this series from scientists across the globe. We first launched this in August of 2020, planning initially on a single 15-week run, but participation and feedback so greatly surpassed our original expectations that we decided to continue. And so here we launch another 15-week series with a fantastic lineup of speakers from St Jude and around the world that span the research spectrum, including mechanisms and biology of cancer, precision therapies, clinical trials, and survivorship research. For those of you who are new to the series, before we begin, I'd just like to take a few moments to introduce you to St Jude Children's Research Hospital. So this is our campus in Memphis, Tennessee, which covers 90 acres and has more than 5,000 employees. St Jude is well recognized as a children's cancer hospital, but not all perhaps are as familiar with the scope of our scientific research enterprise. We have 300 faculty working across the spectrum of basic, translational, clinical, and population sciences. These faculty work in disciplines that range from oncology to epidemiology to cell, structural, and chemical biology. Our mission is to advance cures for pediatric cancer and other important catastrophic childhood diseases. To accomplish this, we're committed to sharing data and working collaboratively with scientists across the globe to help drive discovery and treatments for children everywhere, as I think you'll see in today's talk. Before introducing our speaker, a couple of brief housekeeping notes. If you have a question at any point during the talk, please enter it into the WebEx chat box using this icon and send it to all panelists. And I'll moderate the question and answer session following Dr. Zhang's talk. Dr. Zhang has agreed to stay a little longer, so the Q&A session will continue for a few minutes after the top of the hour at the end of her talk. If you do experience any technical issues, please contact the Science of Childhood Cancer support email address shown here. And so now it's my pleasure to introduce my close colleague, Dr. Jingwei Zhang. Dr. Zhang is Chair of the Department of Computational Biology and the Endowed Chair in Bioinformatics at St Jude Children's Research Hospital. Jingwei has had an illustrious career in genomics and pediatric cancer spanning almost 3 decades. She led the development of multiple computational tools and played a key role in the initial sequencing of the human genome. Upon joining St Jude in 2010, she's played key roles in the St Jude Washington University Pediatric Cancer Genome Project, as well as the parallel NCI Target Initiative, and established St Jude's cancer bioinformatics analysis platforms and pipelines. She's developed multiple innovative cancer genomic tools and has had a leading role in many of the seminal discoveries of the somatic and inherited basis of childhood cancer. More recently, she's led the development of St Jude Cloud, a data hosting, analysis, and visualization platform that's been truly transformative in the field. So Jingwei, thank you, and over to you.
Thank you so much, Charles, for the great introduction, and it is truly a great honor for me to be the first speaker of this new series of the St. Jude Science of Childhood Cancer. As I am a computational biologist, so my presentation will focus more on the data science aspect of the pediatric cancer. So hopefully, some of these the data analysis software component would still be of interest in anyone who is interested in joining the force on tackling how to improve the cure and the survival of pediatric cancer. So I will focus my talk on genomic variants, and my talk will encompassing description of the landscape mapping and application of the results that we derive from research for precision oncology, as well as the data sharing ecosystem that we built. So genomic variant analysis commonly starts with a research cohort, and by performing molecular profiling, we can derive prognostic markers that predict drug sensitivity resistance and others to develop clinical application. And these clinical assays was applied to stratifying patients for precision oncology treatment. On the other hand, as molecular profiling has increasingly become a standard practice in clinic, what we see is that the direction is actually iterative, where the clinical genomics data can be generated and reapplied for research purposes. So I'll start my talk describing our workflow from research to clinic. And this was initiated by the St. Jude Washington University Pediatric Cancer Project, which we profiled major subtypes of leukemia, solid tumor, and brain tumor. In total, we profiled paired tumor normal genomes of 700 patients, whole exome of 1,500, and 1,000 RNA-seq. Importantly, we have experimentally verified over 2 million somatic alterations that really lead to the high accuracy of the findings and lay out the stage for applying these results and analytical pipelines for clinical use. So I want to mention that in addition to publications on Science unveiled by this study, we also published some of the very highly accurate methods that were originally developed using pediatric cancer dataset. And these included tools for structural variation analysis, CREST, copy number variation, concerting, telomere analysis, gene fusion, and visualization tool called Protein Paint. So with these tools prepared and as we move forward, we also collaborate with NCI to initiate the first childhood cancer pan-cancer study. And this is in collaboration with the NCI target team, along with leaders in ALL, AML, neuroblastoma, Wilms tumor, and osteosarcoma. So the study was published in 2018, side by side with Stefan Pfister's group in Germany. And as the Pfister's group has been focusing more on the European sample, we also have offered to share the whole genome sequencing data generated from TCGP to be incorporated in the study. So our work will focus exclusively on the NCI samples. That way, the two studies can reach the same conclusion without using overlapping samples. So in the NCI cohort, we have profiled close to 1,700 patient samples by whole genome, whole exome, and RNA-seq in these 6 pediatric cancer subtypes. I want to mention the whole genome platform that used was from Complete Genomics, so which has the data was generated with multiple iterations of PCR cycles. And that led to the initial report from the company containing about 100 million somatic variants, which we have to apply extensive filtering and manual curation steps, kind of whittling down to 44,000 variants that could be used for statistical analysis, which lead to the discovery of 142 genes. And for some of the variants that were the frequency is too low for statistical analysis, we applied pathogenicity classification and identified an additional 82 additional drivers. So some of the key take-home message that we got from these driver events is that the majority of these drivers are absent in the 3 adult pan-cancer studies. And 62% of these were caused by structural or copy number changes. And these findings really highlight the importance of applying whole genome sequencing for pediatric cancer research. So now let's look at what are the biological processes that were altered in these 6 pediatric cancer subtypes. And we have categorized the percentage of these processes highlighted on the y-axis here with the frequencies on these 6 cancer types. And we have marked these in color, those genes that were unique to pediatric cancer. As we can see that transcription factors is the highest, is the biological processes with the highest prevalence of mutations in pediatric cancer. And the majority of these genes are unique to pediatric cancer, and suggesting that there need to be efforts dedicated to developing innovative therapy targeting alterations that happen exclusively in pediatric cancer. We also performed a mutational signature analysis for the study in collaboration with Ludmila Alexandrov, who is now at UCSD. So one of the surprising findings here is to see the yellow light here was a UV light signature in B-lineage ALL. And they occur in a subset of the BAL, but the proportion of these events are relatively high when you consider the mutation, the overall mutation burdens in these individual patients. And more strikingly, in all of these 8 patients that have the UV light signature, none of them carries the canonical driver fusions that occur in leukemia subtypes. And they are all characterized by aneuploidy in these cases, and none of these are African American. So this really suggests that could be some etiology link of UV light exposure to the increased risk of pediatric cancer BAL. But as our cohort sample size is only 8 for these cases, additional studies needed to really verify if UV light can constitute a non-known environmental risk for B-lineage AL. So this study was led by Xiaotuma and Liu, formerly in my laboratory, and both of whom have started their own independent research lab in collaboration with the NCI target team led by Daniela Gerhardt. And there's also several laboratories from St. Jude participated, and we have collaborated with Lamio Alexandrov and Bob Huder, who was a former postdoc fellow and now works at Tempus Clinical Diagnosis. And the study was supported by funding from NCI. Now I want to switch gear and talk about a different subject, which is drug-resistant mutations in relapsed pediatric AL. And this is a study that was carried out in collaboration with Shanghai Children's Medical Center. So what we have done is to collect patient samples, 103 in all, including the diagnosis, relapse, and germline trios, and profile whole genome and RNA-seq. And these patients were primarily treated using a protocol that was initially developed in St. Jude. So there's the survival rate is more or less comparable to what we see in the United States. So the samples we collected by the time of diagnosis, relapses can be classified into 3 groups. Those that have very early relapse between 9 months, early relapse between 9 to 36 months. This marks the end of the regular ALL treatment, and late relapse, which is post-treatment relapses. So what we have done is looking at mutations that were enriched or exclusive to relapse, and that lead to the finding of 12 genes that are known to be involved in drug metabolism pathways in drugs that are used for treating AL, that including glucocorticoids and thiopurine and methotrexate. So interestingly, when we're looking at the mutational distribution of these genes, we found that the early relapse groups have highly enriched mutations compared with the very early or late relapses, suggesting that therapy treatment may play a role in inducing these mutations that we are seeing here. So we also looked at the mutational signature as the diagnostic samples as well as the relapse samples. In this view, we have marked the y-axis the number of the mutations in each sample at the diagnosis and the relapse. And the samples were sorted by the time to relapse. And we also stratified the proportion of mutations that were likely induced by the mutagenesis processes identified in these cohorts. So again, we were able to replicate the UV light signatures that originally seen in the NCI cohort. More importantly, we discovered 2 novel signatures marked as blue and magenta color here that were only observed in the relapsed cohort. And interestingly, they were only observed in the early and late relapses, but not in the very early relapse cases. And when we look at the driver genes leading to the mutations, we found both the signatures called A and B contributing to the rise of the mutations in PRPS1, TP63, NT5C2, and R3C1. And those are the known driver genes or novel driver genes that we discovered in this study that's contributing to the relapse of the ALL. So the next question we want to ask is that what are the mutagenic agents that can lead to these novel signatures? To answer this question, we performed an in silico experiment by kind of evaluating the whole genome tumor sequencing data that are publicly available, including the pan-cancer PCOG cohort, about close to 2,000 whole genome cases, as well as the NCI target cohort that comprised of both the diagnosis and relapsed cases. What we've seen here is that the novel signature A and B are absent in all the diagnostic cases in both the adult and the pediatric cancer. In the relapsed cases we have analyzed, what we found is that signature A appeared to be present only in ALL and most exclusively in the Shanghai cohort, but the novel signature B appears to be shared between the target cohort and the Shanghai cohort. So this suggests the signature A may be related to some unique therapeutic agents or combination of the therapies that were used in the Shanghai study, while the signature B is likely to be related to the use of the maintenance therapy, thiopurine, that is common for all the ALL treatment. So to experimentally verify that, what we decide to do is to expose cells to the thiopurines and then isolate single cells, cloning them and perform whole genome sequencing and compare with the original the cell lines that were used to look at the new mutations detected in these single cells. So the first cell line model we used is the leukemia ALL cell line model, REH. But the result was quite disappointing, as the reason is that what we see, each of these rows is individual cell lines and single cells that were treated with thiopurines. What we see here is that most of these mutations acquired appear to be shared across these different single cells. So suggesting that they are already present in the initial cell culture that we have. So the treatment is only re-identifying a subclone that's resistant to thiopurine treatment. And indeed, we later realized that the most of the leukemia cell lines are derived from the drug-resistant clones, resistant relapsed clones. So Sam Brady in my laboratory come up with a brave idea and suggesting that using a breast cancer cell line, MCF, which was used for discovery of cisplatin signature, for testing whether thiopurine treatment can induce mutations in this cell line. So I have to say initially I was pessimistic, but the result is actually surprisingly great. So what we see here is the first column is untreated cells. The next two are the thiopurine treated samples. And the last one is cisplatin treated cell lines. So this way that we have a positive control and the negative controls in these all these three cases. So we were able to see very distinct private mutations in the MCF cell lines after the treatment is thiopurine. And when we look at the signatures in these private mutations, first we are able to confirm that the cisplatin signature can indeed be induced through the cisplatin treatment, at least confirming a positive control in our analysis. On the other side, when we look at the clones 3 and 4, which is thiopurine treatment, we see that they do correlate highly with the novel signature B that we detected in our patient sample, confirming that thiopurine is indeed the mutagenic agent leading to the cause of this novel signature be. So from this study, that would suggest that we would like to create a new model that accounts for the ALL relapse. So prevailing model from diagnosis to relapse are the following two. The first is a de novo resistance, meaning the treatment is simply does not have any effect on the tumor samples. And this is not relevant for our ALL dataset because our patients went through remission. The second model is chemotherapy selection. So a subclone present already at the diagnosis persisted to relapse. And based on the calculation of clonal expansion, what we would account to is this will be mostly account for the very early relapses that we observed in our patient cohort. And our new model suggests that could be a two-step process where the first chemotherapy will first select a persistent clone, and then there's chemo-induced mutations acquired, giving rise to these resistant clones that lead to the clonal expansion of the relapsed ALL. And these are this pattern may account for the majority of the early and late relapse cases we observed in our cohort. And I want to mention that the study is a multi-institutional collaboration involving multiple institutions in China, led by Shanghai Children's Medical Center. And on our side, the study was led by Sam Brady, who discovered these mutational signature analysis, designed experimental validation, and also performed some part of the experiment himself. And the clonal evolution study was in collaboration with Ben Raphael's group at Princeton University. So now I want to switch gears to talk about our effort in clinical genomics. And again, this is a collaboration with multiple departments at St. Jude, including pathology, oncology, cancer predisposition, as well as multiple subject experts. So our approach is to perform a three-platform clinical sequencing involving whole genome, whole exome, and RNA-seq. And this was developed through a pilot study utilizing 78 cases. And from 2015 to 2017, we carried out a feasibility study called Genome for Kids, led by Kim Nichols at Oncology. And since 2017, we started to do the real-time clinical service. And to support that, we also included an initial pipeline called Rapid Turnaround Time RNA-seq. And this pipeline was primarily developed to support the total 17 ALL treatment protocol developed by Charles, who has introduced me at the front. And there's also a separate pipeline for FFPE samples utilizing the two-platform exome and RNA-seq. So currently, we have analyzed close to 2,000 cases through the real-time RNA-seq platform. So I want to give a little bit introduction of our ClinGen pipeline for three-platform sequencing, and this is the effort led by Michael Rush in our department. So starting with initial sequencing data from tumor and normal, that we will do variant detection, including SNV, indel, copy number SV, gene fusions, LH, protease, lysine, RNA-seq for tumor samples, and as well as a germline variant for SNV, indel, and copy number alterations. And then we went through a process called variant classification to nominate candidates, which will be reviewed by an analyst panel to finally classify these variants into pathogenic, likely pathogenic, variant of unknown significance, likely benign, and benign. And then the finalized report will be signed out for both tumor and the germline samples. So I want to give a little bit more detail on the variant classification algorithm that we've developed called PCompI, as this has been recently published. and with the code available on both GitHub as well as St. Jude Cloud. So this analytical process started with a VCF file and went through the germline, candidate gene filtering, and also performed the annotation and population filtering, and then go through a process called metal ceremony, which divides the variant into gold, silver, bronze metal, and non-metals. And then we have an interface that allow a panel and their collaborators to provide input and to come back with the final classification interactively. And the platform was developed by Mike Edmondson and Anand Patel, published in Genome Research last year. And this process has been used for the first germline studies published in New England, cancer survivorship germline study published in 2018, and another effort by Lancet Oncology published by Jin Yang's group. And this is an example, as I'm showing here, that utilizing the AL for exome sequencing data in contrast to the genome in a bottle analysis that shows that starting with a huge number, that each iterative process can really whittle down to a very limited variance. In the case of AL, we have like 14 metalled variants, 1 gold, and in the genome in a bottle, 17 non-gold, and panel review gives 1 likely pathogenic. And in the AL cases, this likely pathogenic variant is actually loss of function mutations in ETV6, and it really explains the inheritance patterns observed in this family. So I also want to mention that as we were revising the decompile, we were actually approached by someone in Sweden, Theo Solsi, and asking whether we can incorporate functional data into our variant calculation step. So he has provided us with TP53 functional assays carried out in yeast as well as in mouse. So at least for P53 and a few of the other analysis, a few other genes, we were able to incorporate the functional assays as shown here. The green meant for the no change, and the red is significant, really to enable a more precise germline characterization for some of the, to separate the rare polymorphisms from the functional variant. So we look forward to collaborate with additional scientists who are engaged in doing functional assay screening data so we can incorporate these for some of the variants interested in pediatric cancer. Now I want to mention a few interesting case reports that we kind of were able to use this platform to get some new insight on potential treatment or germline variant association. The first case is a child with metastatic melanoma who had failed multiple therapies. And from our C-platform sequencing, we discovered a novel fusion that involves the MAP3K8 and GNGN2 in frame. And this patient lacked the canonical BRAF V600E mutation and does not respond to any of the therapies, including immune therapy. So based on this finding and the literature search, we recognize that this mutation, the fusion, is likely indicating activation of MAP kinase signaling pathway independent of BRAF. So we recommended a MEK inhibitor. The patient has total response but later developed resistance, and when we sequence the resistant tumor, we recognize this gene fusion event actually has a duplication. So this led us to looking into how common, such a novel fusion event happening in rare pediatric melanoma. So in collaboration with Amita Baharami at Pathology, we performed RNA-seq on 49 FFPE split-way melanoma. To our surprise, MAP3K8 fusion or truncation accounts for the highest, most prevalent event in this cohort. and they are mutually exclusive with all other types of kinase activation events observed in this cohort. And most strikingly, all these fusion or truncations actually led to the loss of truncation in the last exon of MAP3K8, which encodes for an autoinhibitor domain of MAP3K8 based on the previous studies. And we kind of gleaned from the function of these from previous biochemical assays that doing the kind of the deletion experiment that we were able to recognize the role of this domain and the meaning of the truncation. Interestingly, when we look at the exon, such events, you can also get the information from coverage power-ups on the exon 8 and exon 9, and we can see a much reduced coverage in exon 9 compared with exon 8. So using this pattern, we actually were able to utilize the NCI GDC platform by just focusing on the MAP3K8 and looking at the exon 8, exon 9 expression differences, and identify that actually about 7 to 8 adult melanoma samples shown in these red dots here that has elevated exon 8 expression compared with exon 9. And then we just need to have download these 7 cases, perform fusion detection, and was able to confirm the same fusion events as we discovered in the adult pediatric cancer. So currently, we are working with chemical biology department for testing new compound targeting MAP3K8, and this study was published last year in Nature Medicine. So another kind of the interesting aspect of utilizing the whole genome sequencing data is really looking at how they can advance our interpretation of relevance of germline variations detected in pediatric cancer patients. So this case refers to a MUT-YH non-pathogenic mutation G39860 detected in one of the retinoblastoma sample. So we know MUT-YH gene is normally only involved in genetic risk of adult colorectal cancer. And in this particular retinoblastoma sample, we also observed a loss of second allele due to copy neutral LH, as you can see the different VAF between the germline and somatic tissue here. And interestingly, this retinoblastoma sample shown in this color here accounts for the highest mutation burden in the Genome for Kids protocol, and is also among the highest, most highly mutated samples if we include both the PCGT and G4K cases. And when we look at the mutational signature, it harbors a high proportion of mutation that could be contributed by ROS. As MUT-YH is an enzyme involved in repairing the ROH-induced mutations, we believe in this particular case that MUT-YH signal mutation actually is relevant for this retinoblastoma sample as it contributed to the mutagenesis process of this case. So the second case is involving how to classify the pathogenicity of a very rare TP53 mutation, A161, in a ganglioblastoma patient. So in this case, we identified a heterozygous site mutations in the germline, and the tumor sample does not show loss of heterozygosity. But when we look at the RNA-seq, we see the expression is monoallelic. So more importantly, even though the overall RNA-seq expression of this tumor is low, but we were able to utilize the markers, germline variations, polymorphisms in the vicinity or in the intronic region, that we see the whole region exhibits allele-specific expression pattern. So in this case, we were able to use the loss of second allele by some unknown epigenetic mechanism to confirm the biallelic loss of this P53 mutation, and therefore recommend this variant to be pathogenic or likely pathogenic. Now I want to switch gear, talk about our data sharing and visualization on St. Jude's Cloud. And this study was led by Clay McLeod in our department and was published in January, a couple of weeks ago on Cancer Research. So I want to mention why we do kind of cloud versus traditional repository-based approach. Is repository, a centralized repository. It requires each user to be able to have the data storage and infrastructure to reanalyze and reprocess the same data set. While we focus on the cloud platform, data need to be uploaded once to the cloud, and the tools need to be uploaded once to the cloud, and everyone on the global community can all access the same infrastructure on the cloud to reanalyze the data. So as we highlighted at the beginning, our data set actually coming from both the retrospective research as well as prospective clinical study involving both the pediatric cancer survivors, and non-cancerous disease. Currently is only referring to the pediatric sickle cell study. So we have put extensive effort on data harmonization, variant curation, and the metadata connection, as well as expression matrices. And we have developed 3 applications, including one is called PCOM, Pediatric Cancer Knowledge Base, and the Genomics platform that enable people to access data and perform real analysis as a visualization community. And I will give a brief introduction to each of these 3 platforms. And so far, our platform has 100.2 petabytes of raw data from over 10,000 patients. There's over 200 requests that has been delivered with 53 institutions from the world. We have registered users who only want to utilize our tools for cloud-based analysis, as well as close to more than actually 10,000 users per month that access some of the open access data resources we created. So this is kind of the breakdown of the cohort and studies that we have deployed our tools, our data set on the cloud. And I want to mention that this is the kind of what's being put on our publication, and this data accumulation is also dynamic, and most recently we have deployed about over 700 RNA-seq from Charles's 10AL study, and these data are also available for other people to download and also all utilized directly on the cloud for their research. So the real-time clinical genomic streaming is really an institutional effort to ensure that our clinical sequencing data is not only used for patient clinical care, but also can be used for immediate research. So we have developed a semi-automated pipeline called RCTG pipeline for regular data upload from ClinGen data generation to cloud. So this pipeline involves verification of patient consent, QC check, metadata collection, and data harmonization. And we have been, it has been launched in May 2019, and the data has been steadily growing. And I want to mention that even during the COVID, we'll still be able to roll out a massive amount of data to the public. And this January, we just made another major release on delivering about data sets from another 500 cases on St. Jude Cloud. So one of the really unique things about ClinGen is, as every eligible patient in St. Jude gets sequenced, we are able to capture some of very rare pediatric cancers, like some of the blood tumors, germline cell tumors and solid tumors, and many of them can be considered as N1 in our cohort. So that really facilitates the studies of rare cancer types and enable people to access these tumor data as soon as possible. So in our platform that support analysis, we were developing these, what we call the end-to-end workflow. So a scientist without a computational background can utilize these well-tested algorithms to just by doing point and click to launch a sophisticated analysis. So these are kind of just a outline to show you can just select the data, request data access, create a project folder, and also select the tools you want to use and launch the tools, and also select the parameters for running these tools and get your results. So, the tools that we have deployed on the cloud, including some of the public tools, but also tools that were developed by St. Jude's PIs, and also our collaborators. I want to mention that the mutational signature analysis was using the latest and the greatest version developed by Ludmill, and we worked with them to deploy that on St. Jude Cloud, so that people can use the most advanced tools to perform sophisticated analysis using data generated by the user or the data by St. Jude Cloud to perform these analysis. I want to mention two of the tools. One is RNA-seq expression classification, and the other is SysX is a tool developed in my laboratory for non-coding variant analysis. So using RNA-seq to perform t-SNE and looking at tumor subtypes, has been done by many laboratories. So with the advantage for us is we have well-curated tools and curated data sets, and including curation on the driver mutations and gene fusions on individual samples, so that we can evaluate the quality of the cluster based on additional molecular features we have here. So far we've been using 1,500 RNA-seq samples from brain, blood, and the solid tumors. And the interface is highly interactive, and the users basically can select a subset of the tumors for exploration, or selecting samples of interest to look at where they land. And the RNA-seq expression classifiers basically allow a user to import their own sequence and to see where they land on this map. And we found this is to be very useful even for our own clinical application, as some of the samples, gene fusion event is very difficult to detect because the fusion gene is lowly expressed. But by using the RNA-seq expression classifiers, we're able to provide complementary data that support the presence or absence of a gene fusion in these cases. And lastly, I just want to mention one of the tools called SysX that were developed by a former postdoc fellow in my laboratory that was really looking into how to define non-coding variants by utilizing a combined, I'll skip some of these introduction slides as we are running out of time. So our idea is that many of the current non-coding variants were based on the current screen requires a use of large cohorts. So what we focus on is try to use one tumor genome samples, but basically using the RNA-seq as a readout from a DNA driver variant. So what we have done is looking at allelic expression on allele-specific expression, and the model we created is a combination of Gaussian and binomial, which we found that is a better depiction of the empirical data compared with other models we tested. And we also focused on detecting activation events, so outlier high expression has also been used for the first pass of non-coding variant analysis. So the pipeline requires an input data of whole genome and RNA-seq, and to derive the ASC and RI expression. And we also have incorporated additional data set, information like transcription factor motif analysis, TAT domain association to nominate the candidate. So we have performed our testing on the P-lineage ALs because there has already been rich knowledge about the non-coding drivers. So what was really reassuring to us is in a small cohort of 15 PALs, we were able to recapitulate non-coding variants that previously published in 5 different studies just by running this algorithm. And we also discovered 4 novel non-coding variants, including one of the structural variations leading to activation of PRLR, which is a gene involved the JAK signaling pathway. And we found that, based on the deletion event, and this is caused by removal of boundary elements between PRLR and IL-7R, and leading to reutilizing the enhancer elements of IL-7R to activate PRLR. And another event is a novel Tau1 intronic variant that we discovered, and very luckily we were able to get a PDX model from this one of the tumors that have the mutation. We were able to see that this variant actually introduces a YY1 binding sites. We were able to perform ChIP-seq and YY1 ChIP-seq, able to confirm that only the variant allele contributing to the ChIP-seq peak observed here. So finally, I want to give a little bit mention about our visualization tool that was published on January 11th called Genome Paint, and this is a study led by Xinzhou in our department. So, Genome Paint, in contrast to the previous Protein Paint, have a focus of the whole genome instead of only the coding variants. So we have developed the cohort into having the ability to kind of reviewing a cohort view in either the dense mode that I've shown in these lollipop sticks here, or expanded view that incorporate both the sequence mutations and structural variations together in the same view. And we also have the matrix view that can enable people to view multiple variants in multiple genes and perform additional kind of the survivalship analysis through both the variant and the cohort view. And the sample view, which is a genome view for each individual cancer type, is the most advanced version, and as it has rich features, allow people to do detailed analysis on the individual tumor samples. So I want to mention that, some of the use cases we present in the study really demonstrate the power of this viewer. And this is one of the use cases, which is we initially were using the view to see how it can be depicting the known non-coding variants leading to enhancer duplication, like the P-lineage AL has a very well-known NME enhancer show as a power-up of copy number changes here. So as we are reviewing this, we also find that in the B-lineage AL, NAM6 also unexpectedly contain this P-lineage AL specific enhancer duplication. So is this real or not? And the mixed genes right here, 1.4 megabases. And we have high-C data generated from another different study, and we are able to see that actually in this locus, MYC actually do interact with the new enhancer duplications found in NAM6. And so the data set in NAM6, we also generated the phased whole genome sequencing from the previous study. So we're able to kind of confirm the duplication were 4 copies of duplication of this enhancer were coming from one allele, And when we're looking at allelic expressions of the amplified versus unamplified copy, we see there's no bias between DNA and RNA in regions prior to this duplication. There is a allelic imbalance in RNA-seq compared with the DNA in the duplicated region, as well as the downstream massive amount of the enhancer. The RNA-seq expression also show a very dramatic enhancer allelic imbalance in enhancer RNA. As these were the long-range events, so the whole genome is always, even with the phased approach, also have some biases. So luckily, we were able to collaborate with Mark Valentine at Cytogenetics Laboratory, who has performed this beautiful serial RNA DNA fish study that shows indeed monoallelic was observed in 58% of cells. Imbalance was observed in 30%, and only a small fraction of the cells have the biallelic expression of these two loci, confirming what we observed from our molecular analysis using the whole genome RNA-seq data. I will wrap up with some of the new features that we are planning for the St. Cloud visualization community. So we have shown how our data could be used for presenting information that are of interest to people at the genome of the protein scale. What we really want to see is some of our visual graphs could be used by individual users, like do it by yourself with visualization community. So these will be the visual graphs that we're going to plan to support. I just want to show one example called Big Wig, and it's just a two-dimensional X and Y graphs that were used by many analysis, including ChIP-seq, RNA-seq, and all the others. And you can actually import a file from UCSC, Genome Browser, or other places, and incorporate this together with the St. Jude, structural variation data, for example, in osteosarcoma, and the graph is actually a phylogeny conservation that observed from UCSC to gain additional insight in the data set. And then finally, I just want to give a great shout out to all the cloud teams, and this is how we work during the COVID by WebEx, and thank you all for your interest, and please feel free to explore and with the rich data resource and make new discoveries. Thank you.
Thank you, Jingwei, for a truly wonderful talk. We do have some questions. Before going into those, just a reminder that if others have questions, please enter them into the chat function and send them to all panelists, and then I can read them out to Jingwei. So with that, the first question is from Mike Kuoko, I think related to the first part of your talk, Jingwei. Why only filter for coding variants and structural variants? Why not consider variants in non-coding regions in filtering?
Okay, so we did filter for non-coding variants, but we're not using that for driver gene analysis, because the non-coding variants is like 98% of the total variants we have, and it is very difficult. At the time that we're doing the target study, we haven't developed SysX. We're not able to really looking for the what is the proper way to looking for driver non-coding variants. So the non-coding variants were used for the signature analysis for the target. These are only taking the filtered non-coding variants. If we don't filter, we actually will see massive amount of the data coming out from the the target specific signatures, and really will blur the overall profile.
Okay, thanks. The second question is from Madame Babu, actually a two-part question. For the population variant filtering step in your clinical genomics effort, how often do you rerun the pipelines with new data that becomes available? Can you also comment on whether differences in representation of different subpopulations influences the final set of variants that emerge in the pipeline?
Yeah, so the population filtering, we have based on our data set using the NOMAD. So the population NOMAD has already characterized based on the different population frequency, the stratification. So we don't do that in our pipeline to filter directly on each population. But when we were on the phase of the PCOM-PI interaction with experts, is where we got the patient-specific ethnicity. And then we can correlate with the filtering, the frequencies that displayed in PCOM-PI interface to look at what subpopulation it has. So we run the analytical pipeline as a really, really great question. So this is where in our germline protocol, we talked about to revisit the DOSs periodically, every half year to see if there's any additional pathogenic variants that has been discovered through research and other processes that we need to reclassify them. And then in these cases, we will have to issue an addendum to the patients to kind of account for the new pathogenic events that we discovered.
Okay, next question is from Roland Kuiper. Regarding the unexplained silencing of P53 as a mechanism of the second hit, we were able to investigate whether local structural variants like inversion events were involved, with the silenced regions all in a single TAD domain.
Yeah, so in this case, we didn't see any local SV at this point. And obviously, we cannot rule out that could be some other non-coding somatic SME indels that maybe contribute that. So we will definitely looking into that direction in the future. And on the other side, I also want to mention, methylation-based silencing can also be a mechanism. And as our ClinGen data has not been samples has not been subject to methylation array analysis. And I think that really representing a missing component in our current design of ClinGen sequencing. So similar kind of the pattern we actually were observed in the retinoblastoma RB1 by allelic loss, and there's about high fraction of the RB loss second hit was delivered via methylation.
Okay, thanks. You may have answered this question, but from Victoria Hamlin, is the code and pipeline for the non-coding variant identification available publicly?
Yes, I have. It's available both via SainJu Cloud that you can also run interactively as well as GitHub. So I believe I have put in a GitHub link there. And if you cannot remember the GitHub, you can just search my web page. My lab page is also there. So we put a free copy over there. And also, I want to mention the community feedback is actually very important. We have some users also recently a couple of improvements that we could make to further improve that. So if you use the data as a code and see anything that could be further improved, we will continue, making our code better.
Great. So with that, I'd like to close the official event, but as I mentioned, we are going to continue. Jingwei has agreed to spend a few more minutes taking questions, and we do have a few more. So again, thank you everyone for attending, and please stay on and keep sending your questions if you have them. So the next question is a good one with a global perspective, Jingwei, from Naftal Yiri Munsi. Apologies if I didn't get your name correct, but question is, this is a very interesting presentation. The advanced medical technologies help to provide clear diagnosis. What is your advice to the settings and countries with limited medical technologies? How can we be sure we're providing the true diagnosis to our patients?
That's a hard question. So I really, I actually, this is a very interesting question. So we do have some interesting interaction with David Weinstock at Dana-Farber, that he has developed a kind of a small panel, like, 60 gene RNA-seq panel for diagnose lymphoma. So we were looking at whether such a, so the test for these samples, according to him, will cost only $10 per patient, that you can bypass all the pathology tests and go directly to subdivide these types for lymphoma patients. So what we view our role is here, because we have the full RNA sequencing data set generated, So whether we can extract some information to develop these panel-based tests with the best, smallest numbers we can have, so to develop these $10 assays, that will be very good for developing countries like Central America and other places. So this is kind of the the best way I can think about kind of translating our findings from the genome-wide approach to really go down to specific targets and working with individuals, scientists who is interested in developing low-cost assays for developing countries.
Yes, thanks, Jingwei. This is such an important point, and just if I can make a comment, this is an area that the Department of Global Pediatric Medicine is working on very actively with pathology and others, thinking about the right sort of assays to deploy around the world to improve diagnosis and management of childhood cancer. So I'm not sure which area you're from, Naftal, but please get in touch with us if you'd like further information. So thank you for that question.
Yeah, Charles, if I may, I just also want to mention something else that we are thinking. So, we were hoping to get, so the other one is, obviously computational cost in cloud can also prevent some people from doing some extensive analysis. So at this point, we don't have a good solution to solve this problem, but we are interested in kind of in the future to explore is there any way that we can help, work with tech companies to reduce the cost of computing for data generated on the cloud. This including having utilized some queues that allows for low priority queues that takes longer to do the computing, but the machines are a lot more cheaper than what you do kind of the state of art computing. And the other is there any general way that we can approach this problem of getting support of reduced or even free computing on Hsinchu Cloud. So this is a question that I'm very, very interested in explore of how do we facilitate research and clinical applications for regions that are not as resourceful as we are.
Yeah, great. So thanks for that. And next question is from Nuria Lopez Bigas. Great talk. The novel signature A and B associated with treatments is very interesting. I'm wondering if you have any experimental evidence of novel signature A and etoposide.
We don't. So initially we thought etoposide is a potentially good candidate because the dose used in Shanghai is somewhat different from what's used in COG. But we tried that and was not successful, and so this is where the problem is getting more hairy. I'm looking at some of the combinatorial dosage, different dosage that might be used in the Shanghai protocol versus in at the COG protocol. And I want to mention that the novel signature A actually is not a signature pertaining only to Chinese patients. So this is why in our recent paper for the cancer discovery paper on the Hsinchu Cloud, we were able to apply the signature analysis on our AL samples and found the signature A was also found in a St. Jude patient of Caucasian descent. So we were pretty sure it's not a ethnicity-based observation, but a therapy-related event.
Thanks. Next question is from Hongmei Zhang. In addition to tumor status and genomic data, do the data also include other information such as potential risk factors?
We don't have explicit potential risk information, but we did include what we call the biomarkers status there. So these are the biomarkers that really usually have some of the prognostic or diagnostic value. So for example, we do include for samples of BRAF mutation status in melanoma and MYC amplification status on neuroblastoma samples. So we decided to do this annotation or gene fusion status in rhabdomyosarcoma. So for these are the common biomarkers used for for risk stratification, and we found it's useful. And this way, instead of having everyone users have to download the data, RNA-seq data, perform gene fusion analysis, we have these key features already annotated in the sample meta sheet. So scientists, if they only want to find the fusion positive cases, they can just grab these samples and do these, instead of to do the extensive analysis to get the fusion status or big amplification status from our data set.
Okay, great. Well, that's all the questions we have, Jingwei. So thank you again for a wonderful talk. Thanks for staying after the hour to take some additional questions. And thank you to the many, many people who have joined today. And we look forward to seeing you again next week. So thank you, everyone.
Thank you, Charles. Bye.